Okay, so the rest of this unit is protozoa. It's divided between intestinal protozoa, blood protozoa, blood and tissue protozoa, and then protozoa from other body sites. So it kind of breaks it down into where we're going to find these. There are a few in this chapter that we will identify possibly in the clinical lab because of some rapid tests, some rapid immunology tests that are involved with those. Some of this was in that last chapter, the unicellular eukaryotic organisms. They have specialized organelles, multiply by binary fission. These are some of the significant ones, are the clinically relevant. Again, in the United States, we do not have a lot of parasitic infections, and we're going to see why the few that we do have to do with either lifestyle in this unit or with unprocessed water, things like that. The 5 groups these are divided into are amoebae, flagellates, ciliates, sporozoa, and microsporidia. Some of this has to do with their motility locomotion, it has to do with their shape, it has to do with different characteristics. So the intestinal amoebae is what we're going to start with. They have pseudopodia. These are like little false feet. It's like the organism has little projections that come out. So pseudopodia means false feet. They're free living, they can be pathogenic or they can be non-pathogenic. We're going to talk about a lot of these amoebae that are not pathogenic. They're in the intestinal tract mostly, so they're GI organisms, but they have been found in other body areas. Because they have the motile trophozoite stage, you can occasionally see these in the stool. That's why those fecal specimens I have down there have very few of these. Because the trophozoite stage is very fragile. It breaks apart very easily. Yes, Kara?
What does free living mean?
They can live in water and in the environment. You remember we said, I'm sorry, I should have clarified that. Remember we said that parasites are generally dependent on a host, but they can be in some forms, in some stages, free living. There are a whole group of amoeba that live out in the environment. We're not going to study those. There's all sorts of water amoeba out there that if you took that appropriate biology course, you'd probably study all of those. But we're not going to concern ourselves with these, but some of the ones that do affect humans can live in a water source or in the environment. Giardia that we're going to talk about, that is one of the more common infections here in the United States. Even though with purified water we don't see it, they live out in the water. They can live in chlorinated tap water. They survive, actually, them and Cryptosporidia, they have to be filtered out, and we're going to get to that. So this is showing some of the structures. With the amoeba, we mostly recognize them based on kind of number of eyes. These are little inclusions, and we call them little eyes because they look like little eyeballs. So that's one of the features. This slide does not have the written information, but if you look at the figure 47.1 in your book, 1, 2, and 4 are blastocystis. Then it's showing the nucleus right here in the macrophage. This is showing 9, 10, and 11. I'm sorry, I can't read with these glasses. Are showing those. 12 and 13 are actually leukocytes. 14 is pollen grain. 15 is a pollen grain. 16 is what's called a Charcot-Leyden crystal, and we're going to talk about those in just a minute and what those are. When our body is exposed to a parasitic infection, our eosinophils increase. Now, I may have already studied that, I don't know. And these crystals can be produced, so that can be significant even though we don't see parasites, because if you see those crystals, it indicates that eosinophilia has happened. And it could be other things, but possibly if you suspect parasites already. So those are things. I do have a sheet. It is very old. I'm going to try to find a better one with what we call oh my goodness, what do I call that sheet? It's junk. It's basically junk in your stool. Like I said, these are pollen grains. Well, why are pollen grains in your stool? If you're walking around during pollen season and you talk outside, guess what? You're swallowing pollen. If you're standing around eating outside, you know, you're breathing in pollen, you're swallowing it, and you can see the pollen in the stool. You can see those shapes. You can see vegetable shapes. So our stool is full of all of that breakdown of food. So you're going to see all sorts of things. A lot of things, these are round. You could confuse it with a parasite. So there's all sorts of debris and junk, and I do have pictures of those. Okay, our entamoeba, most of these are not pathogenic. One that is pathogenic is Entamoeba histolytica. A lot of the others, you have to depend on the signs and the symptoms. If you actually do identify those out in the stool, a lot of times the doctor has to decide are they pathogenic or not. And a lot of those are mixed with other things in the stool also. Some of those other entamoebas that are not pathogenic. These are worldwide, so it doesn't matter where you live. As we study parasitology, I'm trying to get the seat down. As we study parasitology, we're going to see that some organisms occur more in certain geographic areas, but some are just worldwide, that they don't care where they are. This one, actually, industrialized countries seem to have more of a risk for these than underdeveloped countries. You can have amoebiasis, it's called, and you may not have any symptoms. You may have severe abdominal pain, frequent bowel movements, tenesmus. I meant to look up tenesmus. I do not know what that word means. If somebody wants to look it up for me. I had a whole list of words I was going to look up last night because this was in their PowerPoint and I almost took it out. It can cause a very invasive disease. This is where it gets into that mucus lining of your colon. You know, we talked about that in the invasiveness of certain bacteria last fall and how they affect our intestines, but if they get into that lining, they're going to cause colitis, toxic megacolon. Your colon increases in size. Dysentery, another word I never heard was trogocytosis. Did somebody look up tenesmus? What does it mean?
Feeling that you need to pass stools.
Okay. Frequent bowel movements and feeling that you need to have that bowel movement. So we all learned a new word today. They can cause other problems. They can be invasive. They can get outside of our intestinal area to other parts of our body. They can go to our liver and cause abscesses. They can become metastatic, spread throughout our body.
Trogocytosis is when a cell nibbles another cell.
A cell nibbles another cell.
Nibble?
Okay.
Mm-hmm. What is?
Trogocytosis. Okay. Well, it was in parentheses. I thought it was just another name for dysentery, but the cytosis, yeah, a cell nibbles another cell. Okay, so we've learned 2 new words. Okay, so, and I've also got a sheet for y'all I need to post that talks about the different life cycle stages, but we have the cyst stage, we have the oocyst, we have trophozoite stages. The cyst and the trophozoites can be found in starpasta in the stool. The cysts are very hardy. We talked about cyst forms of bacteria back in the fall. Well, parasites can have cyst forms. This is where they can form, the bacteria form more what we call spores, but it's very similar. When they form the cyst, it helps them survive out in the environment. How do we get this? We ingest the mature cyst in food or water or from our hands that is fecal contaminated. It can also be gotten through sexual contact. So again, here in the United States, we are a little better about hygiene, and you see, it's kind of disconcerting, but you go in any restaurant or fast food and as employees must wash their hands. Like, do we not wash our hands anyway? But, you know, at home you have to be careful. You're taking care of your child. You know, you change a diaper, you got to be real careful that you wash your hands well before you walk into the kitchen and start cooking. Huh?
Or get your dog.
Yes, most daycares have gone to where they wear gloves to change diapers, and I'm sure hopefully they're washing their hands anyway, because as you probably learned here, even though we wear gloves in the lab, we still need to wash our hands, because you don't know if that integrity breaks down, or you taking the gloves off, you get something on your hands. It is usually, it can be extraintestinal, but it's really not considered invasive. The trophozoite can stay in the intestinal lumen, and you can be an asymptomatic carrier. Then you would pass the cyst in your stool. So it just kind of hangs out in your body, but the trophozoites, the motile stage, form cysts. You pass them in the stool, then you can make somebody else sick, even though you may be asymptomatic. The trogocytosis, which we just talked about a minute ago, is where it gets into that intestinal mucosa. And then if they travel through your bloodstream to other sites, that can be dangerous. And again, I don't know why I started to take these out, but y'all said y'all didn't care yesterday, asking lab. It put in life cycles for some and not the others. I just left it intact how it was. But it shows ingesting fecal contamination. It shows it going through your colon, through your digestive tract, can be self-limiting if that's where it stays, but then it shows it being invasive, going to your brain, to your liver, causing other problems, even with your heart, so it can be quite serious. So the agent, it is called amoebiasis, or amoebic colitis, or amoebic liver abscess. Those trophozoites, we've said that yesterday, or in the lecture I recorded yesterday, that a lot of our protozoa are characterized by whether they have motility or not. Okay, so these trophozoites are motile. The amoeba are motile. They're very rapid movement. They move very, very fast, and they don't really have a direction. So they just kind of dart out and move out in all sorts of different directions. It's not like a planned pattern of movement. They have a finely granular cytoplasm. They have red blood cells in their cytoplasm when you have dysentery. So all of this, I guess that's where it eats other cells or bites other cells. This is showing some of these. Not a great picture. The CD website has a much better picture of this. So in the cyst, we have 4 nuclei. Again, we do not have micrometers here in our lab, and most hospital labs do not, but in the reference labs, they're going to have that to measure these, to see the size. They have a little bar inside called a chromatoidal bar with blunt or rounded ends. If you go back to that figure 47.3, it shows those bars. Also figure 47.3 is on several pages. On page 650, if you have your book, right in the middle, it shows the nucleus, the karyosome, and it shows those little chromatoidal bars. I should have seen if I could put this picture in, but it does have the different groups. So this one, I do have pictures I'm going to give y'all that show these, but it shows the different entamoeba. The trophozoite, and it does show the trophozoite also in this picture on page 650, pictures 1 and 2. It shows the trophozoite because it's elongated, because it does move. It does have a nucleus. The cytoplasm looks like ground glass. It's very, very fine, and it fills in the whole thing. 15 to 20 micrometers, and it may have those ingested red blood cells because it consumes and eats those red blood cells. What is some of the problem with the disease process that harms our body? It is ingesting red blood cells, and in doing that, it's doing what? It's lysing those red blood cells. It causes tissue destruction. It lyses the host cells in our body. It lyses other cells. It destroys tissue. We already said a minute ago it can be asymptomatic. If it's asymptomatic, they're not going to have those red blood cells when you see it. They're not going to engulf those. But even though you're asymptomatic, it is still at a risk for spreading in your body and becoming invasive. If you do have the intestinal disease from this, you have those trogocytosis, bites out of the cellular membranes. You can get ulcers, what's called colitis, dysentery, abscesses. If it gets to other parts of your body, of course, we already mentioned the liver and the metastatic, how it spreads. This is diagnosed by an O&P. We discussed how to collect the O&Ps yesterday. So the O&P specimen is collected. You can report Entamoeba histolytica / dispar group is another variation of just Entamoeba histolytica. You do report the form. If these are reported out, they're going to report if cysts or trophozoites are seen. It's not important to quantitate to get a number. So as long as the reference lab, research lab, whoever's reporting this reports the presence, number is not, doesn't make a difference in how it's reported, the extent of your illness, or anything. There are antigen detection tests. This is where we take the specimen. We have antibodies as our test methodology. We look to see if the antigen is present. There are histology tests. This will be done in the pathology area of the lab. They would take the stool and do different stains than we would do in the clinical lab. There's some molecular tests, nucleic acid based, real-time PCR, and serology. The patient's blood can be drawn to see if antibodies are present, so there is serologic detection. Even if you're asymptomatic, and why would you be collecting a specimen if you're asymptomatic? There could be other things just going on in the digestive tract, and maybe they just do an O&P just to see, because I told you a lot of doctors do that. And maybe there's not symptoms of this type of infection, but it still would need to be treated. I told you we weren't going to concentrate on drugs. A few of these slides had medications. I left them in here. But there are medications, anti-parasitic drugs. You know, when we talked about our medications or our antimicrobials in the fall, we concentrated on antibiotics, which are antibacterials, but there are anti-parasitic medicines, antifungal, antiviral medicines. And as far as life cycle, yes, we as humans are reservoirs. We can give it to other people or even animals. If the trophozoites or cysts are in our stool, in the environment, in the water, and animals can get this from us. So how do we control it? Sanitation, wash our hands, be careful with food preparation, things like that. For our non-pathogenic intestinal amoeba, these have what's called a non-progressive motility, where the Entamoeba histolytica darted and moved around. This just doesn't go anywhere. It has a little bit of motility or movement, but they don't really go anywhere. They are also worldwide. Again, generally are non-pathogenic. Occasionally, on rare occasions, have been considered a cause of illness, but as I said earlier, they may be present with other pathogens, other pathogenic parasites. One important factor, if we do identify these in stools, is to look for a contaminated food or water source. So even though they're not causing us a problem, we got it somewhere, which just means we should look at our food and water sources. These are all reportable to the health department if they could be present in our water supply. Okay, so for the non-pathogenic, Entamoeba coli, 15 to 50 micrometers in diameter. It's a little bit bigger than the Entamoeba histolytica. Already said the motility doesn't really go anywhere. It's very sluggish. So it's going to move. The pseudopods are going to come out and cause a little movement, but it's not going to travel very far. The nucleus is a little bit different. The chromatin, instead of being the bars, distinctly like the histolytica, they're going to be a little more clumped and irregular. I can't find it. And that was Entamoeba histolytica. I'm not sure which one is Entamoeba coli, 10 and 11. If you look on page 651, you can see that the bars are not as distinct as they are in the Entamoeba histolytica. The cyst may be elongated. With our Entamoeba coli. One way to identify it, you have 8 or more nuclei. It has the most nuclei of any of our Entamoeba. It can have greater than 16. And I'm not sure why there's a star there. The karyosomes, the chromatin is a little bit different. And again, those chromatoidal bars are clumped, irregular, and may even have a splintered look. Trophozoite with our Entamoeba, I keep want to say Hartmanni, Entamoeba histolytica. We said that cytoplasm in the trophozoite was very fine, like ground glass. In the Entamoeba coli, it's very coarse, has some vacuoles in it, which are hole-like structures, so it does look different. Okay, so they're considered non-pathogenic, but you get them from contaminated food or water. Again, you often have a pathogen and a non-pathogen together. May not be able to identify these morphologically. It is dependent on seeing those little inclusions or eyes, and sometimes if they've broken apart in the stool, you may not be able to see that, so it's very hard. For definitive ID for these non-pathogenic ones, a lot of times we do use immunoassays or molecular nucleic acid testing. Another one is Hartmanni. It also comes from contaminated food or water. A little bit smaller, definitely, than the Entamoeba coli. Looks a lot like the histolytica. Does have the chromatoidal bars, a lot like the histolytica. Again, we look at size, immunological testing, and nucleic acid detection, because this one is a little bit smaller. That chart 17 and 18 on page 651 is the Entamoeba Hartmanni, and it is quite a bit smaller in size. So again, we said it looks a lot like the histolytica, but smaller. Has the 4 nuclei. That chromatoidal bars are very similar. Trophozoite is very similar also, has the finely granular cytoplasm. There's Entamoeba polecki and gingivalis. Polecki is more associated with pigs. The gingivalis doesn't have the cyst stage. We're going to learn with some of our parasites, some have trophozoite stage only, some have cyst stage only. This one only has this trophozoite stage. Has been isolated from lung infections, from the mouth, and from the genital area. So a few different areas. Endolimax nana. This is another amoeba. Again, generally non-pathogenic. We find in contaminated food or water, though also very sluggish motility. This one, the only way to definitively ID it is to do a stain smear. So you're not going to see it generally in a wet mount stool specimen. You're going to see it in a stain smear. Here's all the cyst information, the karyosomes. If it is an unstained wet mount, you would not see the nuclei at all, so it wouldn't help you on determining that, those little eye nuclei. Usually you can't see the chromatoidal bars either. Trophozoite, very vacuolated, very granular, which is similar, the Endolimax nana, to our Entamoeba coli trophozoite. Through the cytoplasm and the trophozoites of the Entamoeba coli. Sorry, I'll get it out. Another one is called Iotamoeba bushleyi. This one has worldwide distribution. Again, poor sanitation, non-pathogenic, very active motility. Looks like the Entamoeba nana, the trophozoites, and you can't really differentiate the two to the species level. Okay, another one, Blastocystis species. The book just listed it as species, but I threw in the name hominis because that is one of the more common species that you may see written in literature and what was more in my older books. That whole list we've just talked about, except Entamoeba coli, except Entamoeba histolytica, was non-pathogenic, right? Why were we studying them? Because they can be in contaminated food and water. And even though they're generally not pathogenic, depending on your immune system, they can be, and you want to know if your water is contaminated with those or your food supply, because you do want to clear that up. This one does have some pathogenicity, they think. It's very controversial. There have been people that had diarrhea. They could not figure out what else was going on, but there was large numbers of these. So they're thinking it can be a pathogen more than they used to think. Causes cramps, nausea, diarrhea, fever, vomiting. This one is very unusual in that it was once classified as a yeast. A yeast is a fungus, not a parasite. Its makeup, the characteristics of it, the way it reacts in testing, it was put in the fungal category and in the yeast part of the fungal category for a very long time. They've now changed it to an amoeba. It does have the pseudopodia like the other amoeba, that it extend and retract. It ingests bacteria. It doesn't have a cell wall. They're still studying it though. It's still very controversial on where it should be placed and what category it should go into. It has 4 forms. It has a cyst, thick and thin-walled, a central vacuole. It's amoeboid. It's granular. You can see it in the stool. You can do immunology testing, ELISA testing, and fluorescent antibody test. You can do antigen detection with the ELISA and antibody detection with ELISA and fluorescent. So you can check the stool for the antigen or their blood for the antibody. Blastocystis species. Again, this is still including the hominis where I threw that name in. Because they don't know a lot about it, because they can't even figure out the taxonomy well, they don't even understand the life cycle completely. They don't understand the transmission completely. They do know that the cyst is excreted in the feces. You probably get it with contaminated food or water, touching surfaces, fomites contaminated with it, or through auto-inoculation, fecal-oral route, which is how we contaminate ourselves. We don't wash our hands. We're not contaminating others. We're contaminating ourselves, possibly re-inoculating ourselves. They were classified as amoeba because the way they develop in the digestive tract, the way they travel, the way they multiply is more like an amoeba than a yeast. This one, because they're not sure of the pathogenicity, they do quantitate the numbers. So they do note how many are present to determine if it is just present for some reason or if it is possibly causing an infection. Here's the cyst size and the shape and the trophozoite. We rarely see the trophozoite amoeboid form because it pulls its pseudopod in. It's hard sometimes to see that trophozoite form and see those pseudopods. Our next group are the flagellates. The flagellates have flagella to make them move, where our amoeba had the pseudopodia, the false feet. The flagellates have a flagella. They can live in the GI tract, the bloodstream, or tissues. Common ones are Giardia duodenalis, Dientomoeba fragilis, Chilomastax meslei, and Pentatrichomonas hominis. In the flagella, can look a little bit different. We're going to see from the picture. Sometimes a flagella is a single kind of thread-like organism. Sometimes it's sort of a almost like a flag-like, you know, those flags you can get that you hang up that kind of do like this, that kind of wave to the side. It kind of looks like that. It's called an undulating membrane. That's a bad description, but we'll look at those. So Giardia duodenalis. For years, this was called Giardia lamblia. Then they started saying Giardia intestinalis, which included lamblia and duodenalis. This book calls it duodenalis. I've thrown in these other names because I don't know if boards have caught up with that word, so it's all really the same name for this, for Giardia or Giardiasis. Cysts are very, very hardy. This is one that can survive for months in cold water. It can survive in chlorinated water. You can find both the cyst and trophozoites in our stool. These have the sucking portion, so the trophozoites can attach to the mucosal lining of our intestines by this sucking disc. So what happens is we ingest the cyst in contaminated water, food, fecal-oral route. This one we've said can survive in extreme water temperatures and chlorine. This is why our municipal water systems not only treat our water with chlorine, fluoride, whatever they treat it with, it tastes horrible to me, but they filter it. This is one I mentioned, I think, in lab, this one in Cryptosporidia, that if the water's not filtered, can infect us. That's why if you're out hiking, camping, you don't just drink from a stream. There's tablets you can get for backpacking, camping, but they don't get rid of Giardia and Cryptosporidiosis. You have to filter your water. So again, water supplies have to be checked. It can be gotten from well water that's not filtered. People that still use well water. Can occur in recreational water because it can be in the environment. You can get infected from recreational water. Sometimes there's outbreaks in daycares because of that fecal-oral route. It's one of the reasons daycares have gone to the wearing the gloves a lot of times. More prevalent in warm weather, more prevalent in children. Okay, the rest of these slides are just going to say duodenalis, but we're still referring to it as lamlia or intestinalis. You can be asymptomatic. You can have acute symptoms, diarrhea, abdominal pain, vomiting, nausea, bloating. It can become chronic. You can have malabsorption. What is malabsorption? This is when your food is not absorbed in your intestines. You have to have some food absorbed as you eat it before it goes to waste in your stomach and intestines. There are some antigenic variations. So some of the Giardias have different surface proteins, but those can be detected and still it can be identified based on that. But they have discovered that they do have some variant proteins as part of this genus. How do we identify it? And I wish they had put these pictures on the slide, and I should have. We'll look. There's quite a few pages on the entamoeba. This book has more about entamoeba than any I've ever done. Usually I don't go into that much detail. Page 663 is where we have our Giardia. It is a oval-shaped cyst. It has 4 nuclei. In the mature cyst, the immature may only have 2. It has the little flagella fibrils are visible in stained wet mounts and in stained smears. The trophozoite is pear-shaped, has 2 very large nuclei, has the sucking disc and flagella that you can sometimes see. Here's the picture. Yeah, it is in here. So this is the cyst form. You can see 2 of those nuclei. I can't see 4. This one you can see 4. You can't really see the flagella. This is the trophozoite. You can't see real well, but these are the flagella coming out. You see those 2 large nuclei. This one is described as, it may be on my next slide. This one has descriptions called owl eyes or old man with glasses. You see it's bisymmetrical, so you see the one eye on each side. So very, very distinctive in the trophozoite form. Not as distinctive in the cyst. This will be one that we see in our practical in that trophozoite form. Not in a specimen, but in a picture. So this one will be one. And again, I'm going to give y'all a list, so don't worry about that. But I will give you a list. It does show a side version picture in the book that shows that sucking disc, but you can't see that really in the specimen. So it is found in stool exams. You can also do antigen detection, histology, nucleic acid. Because there may not be many present, and because the trophozoite is the easily identified form, and trophozoites tend to break up in the stool specimen. It may take several stools. I don't know if I mentioned yesterday, but if a doctor wants to check for ova and parasites and do a study, they're going to ask you to collect 3 different specimens on 3 different days. Just like with our mycobacteria, we usually collected 3 sputums or 3 bronchwashes, things like that. Excuse me, to make sure that you can isolate and identify those. Another one is called Chylomastix mesnilii. This one is worldwide. This one is generally non-pathogenic also, but it does indicate contaminated water. You can get it from contaminated food, water, fecal, oral route. The picture for it is on the top of that same page. It looks kind of like, to me, like an upside down giardia, where the flagella end of the giardia was down at the pointed end. The flagella is at the fatter end of this, and there's only one eye, if you want to call it that, one nuclei. So it looks a little bit different. It's considered more lemon-shaped. Has a knob on it at the top where the flagella are. Sometimes we'll have a little crook, a little fibril crook called a shepherd's crook. The trophozoite is more pear-shaped. That's the one that looks like the upside-down giardia to me. Another one, Dientomoeba fragilis. This one, a lot of times it's found in children. They'll complain of intestinal symptoms, diarrhea, nausea, vomiting, tiredness. This one, they're not completely sure of the route of transmission, but it has been found along with pinworm infections. So they think it is transmitted inside the helminth or pinworm egg. And we'll be talking about helminths in the next unit. We talked about briefly in lab about pinworms yesterday and how we collect for those. So sometimes, since it is found with that, they're pretty sure it's possibly transmitted that way. So it is inside that egg, and one parasite is inside another parasite, and that's how it's transmitted. Rarely see the cyst form in specimens. The picture of that is on, I think it's on that page. I'm sorry, I can't see. It may not be on that page. We have to look at our CDC website. Usually can't see the flagella. Another one, Pentatrichomonas hominis, is worldwide also, non-pathogenic, non-invasive, again in contaminated food or water. Again, it indicates, again, why are we so concerned with these? If they're present, that means the water is fecally contaminated somehow, or our food source. Well, if it's fecally contaminated with these non-pathogenic, it's possible that a pathogenic one is there as well. So it's always notable if you find them, hey, they may not be a problem, but let's look really carefully for some of the others that can be pathogenic. No cyst stage, very similar to our other trophozoites in this section. Our next group is called the ciliates. Instead of having the pseudopodia feet to move or the flagella, these have little tiny hair-like structures called cilia, generally on the whole surface of the cell. So where the flagella might be 1 here, 2 or 3 here, or that undulating membrane, this is going to be completely surrounding the cell. Has 2 types of nucleus, a large nucleus, micronucleus, and usually has more than one. This is the only ciliate that is pathogenic to humans. Neoballentidium coli. I want you to write to the side somewhere or note, and I don't think I put it on a slide, no. The old name for this was just Ballentidium coli. So if you see the word Ballentidium coli, it is the same thing as Neo, okay? I do not know why they changed the name. I looked up, and I think it tells in our book that there's a discrepancy in a lot of literature. Some still list it as Ballentidium. Some people list it as Balanatitis coli, and some listed as Neobalantidium. This book listed as Neobalantidium. Previously, Balantidium is how they list it. So just think about that neo. If you see Balantidium coli, it is the same thing. All of my pictures are going to say Balantidium coli, and this one is included in our practical also. Found worldwide in the tropics. It's associated with pigs and monkeys. We get it contaminated food or water. The trophozoites go through what's called encystation to produce those cysts that are invasive, that are infective in our stool. This one can be very asymptomatic. We don't know we have it, but it can also cause diarrhea, abdominal pain, all of those things, and be very severe if you're immunocompromised. The cyst is fairly large. It's one of our larger cysts. The trophozoite is large also, but it has those cilia all the way around. Our next group is called Sporozoa. With our Sporozoa, they can be in the GI tract throughout life. They form cysts, sarcocytus, toxoplasma. These are worldwide also. This one, Cryptosporidium, we did not know about till the 1970s, but it is one of our more common waterborne diseases. If we have HIV patients that present with diarrhea or intestinal problems, the number one organism we find that's a parasite is going to be Cryptosporidium. Treatments based on the symptoms. You need electrolytes, you need fluids, management of any pain. This one, they have found that it can possibly be inhaled, but usually we get it through the contaminated water. The spore and the oocyst are in feces, animal feces. They have found outbreaks in unpasteurized apple cider that got contaminated from cattle feces. Because it is in the water, this one compares very closely to Giardia, but Giardia is a flagellate, and this is a sporozoan. They're both found in water, they're found in the environment, they both survive chlorinated water. So we can find it out in the environment from our water. There was a very large outbreak in 1993 in Milwaukee, Wisconsin. The city water system was compromised and the filters messed up. So the chlorination, fluoridation did not work. The filtration whole system went out on the filtration part, and over 400,000 people were infected with Cryptosporidium during that year. So it was a big outbreak. So they're resistant to chemicals, even bleach. This one is hardier than the Giardia. This one can withstand some bleach and chlorine. If you're going to clean with bleach or chlorine with this, you need to have a dwell time that's a little bit longer with your cleaning product, so they can live quite well. Again, may be in some asymptomatic, may have watery diarrhea. Again, very severe in the immunocompromised. So with the Cryptosporidium, again, we want to be very, very careful. I don't think I include Cryptosporidium in your practical. I'm going to have to go back, and I don't have Dientamoeba fragilis. So I'm going to have to go back and look at that. The life cycle for the Cryptosporidium is in your book, and of course it's going to be on the in the CDC website. Oh, it is on here. I couldn't remember which ones were left in here or not, and which ones they put in the book. This shows the life cycle, shows the animals, the environment, lakes, streams, and how that whole process works. So we can see the oocysts, the sporozoites, and yes, I had some descriptions written down for y'all, some definitions. I will get that for y'all that make a little more sense on these. We can see them in wet mount. We can see them in permanent stains. This is showing a picture of the Cryptosporidium in a stool specimen and in tissue. this particular slide, and I meant to change it. It's called laboratory diagnosis and treatment. And it said diagnosis, but then this is the diagnosis at the bottom. And I meant to go back and correct that. I don't know why that's so crazy. Laboratory diagnosis should be the modified acid-fast stains, antigen detection, PCR, and histologic. But that was at the top, and I went back and forgot to change it. Drug of choice, nitazoxanide. Another sporozoan, Cyclospora catanensis, waterborne. This one, though, has been found in produce. Generally, though, is if you don't wash, if you don't wash. The problem with this one is you wash your produce, but if the water's contaminated, it doesn't matter. You're washing the produce with contaminated water. Produce that it has found more commonly in for some reason are raspberries, basil, and lettuce. You consume those oocysts, and then those are shed in the stool. This is why it's important when you travel to other countries that we don't know if their water is processed correctly. You don't want to eat fresh fruits and vegetables from a market if it's not washed in good water. You have to be very careful about that. Same kind of symptoms as all of these, intestinal symptoms. This one can sometimes have a relapse once you think you're better. There are stains that detect this. There's ultraviolet fluorescence, nucleic acid detection. This one looks a lot like the cryptosporidium, the cryptosporidiosis, but they're 2 times larger. So this is where I told y'all the micrometer is very, very, very important if you're in a lab that identifies these, because it's going to look the same, but you would have to measure and find out the size for these. Most of these, I know Giardia, and it wasn't listed on the slide, I apologize. Giardia, Cryptosporidium, and Cyclospora are all reportable to the health department, to the local health department. Some hospitals report all parasitic infections that can be in food or water, but those 3 especially are reported. Wash your produce in clean water, very important, not just any water, and wear gloves when gardening. Another one is Cystiospora belli. This one's more in tropical areas, affects more people that are immunosuppressed. So not as common. You have acute diarrhea, but it's not bloody. You have cramping, can last for many weeks. This one, a lot of times that eosinophilia is present. Even though our eosinophils increase in any parasitic infection, they are especially prevalent in this one. You can examine fresh material, various stains, histology specimens. This one actually, an antibiotic is the drug of choice, not an antiparasitic. Trimethoprim sulfamethoxazole is an antibiotic. Sarcocytosis, there's Sarcocytosis hominis, who has an intermediate host. We've talked about some intermediate hosts. We're going to see some other organisms, especially our helminths, that have a lot of intermediate hosts. This one sometimes has an intermediate host of cattle. The Pseudomonas has an intermediate host of pigs. This one is not going to be water ingestion or contaminated water, but if we do not cook the meat. If we consume the meat that has this in it, then we can have muscular problems. We have muscle tenderness, weakness, bronchospasms, fever. If it does affect our intestines, it's a lot of times asymptomatic, but can cause regular intestinal type symptoms. Because more commonly you have it in your muscle from consuming the meat, then to determine if you have it, they have to do a muscle biopsy on you. They have to go in and pull out some of that muscle. Do some different stains, things like that. If it is in the intestine, they can look to see it in the stool or do molecular testing. So we have to cook our meat. I know some people eat more undercooked meat or raw meat, and this is like salmonella. Every chicken and egg doesn't have salmonella, but we want to make sure we're assuming it does. But if meats are processed, if the farmers are checking their animals, which they're supposed to do, then hopefully that's not going to happen whether we undercook our meat or not. The last group is Microsporidium. These are worldwide also. This particular group has a lot of characteristics of our fungi, which are prokaryocytes, not eukaryocytes. We get these from inhalation or ingestion or from contaminated water. There's a whole list of some that cause diseases. These are not seen a lot. They're pretty rare. We get them from other humans or from animals, from the spore. These are usually found through stains, histologic stains, molecular testing. I can't talk today. There's different forms of therapy. A lot of times these are associated with people that are immunocompromised, that have HIV, and here's a list of drugs. Very important in the healthcare setting. Okay, when we are reporting intestinal protozoans, this is the whole group we've just covered. We of course say no parasites noted if we see none. If we do see them, the body forms are present. Try to report the correct genus and if possible the species. Those charcoal-laden crystals are reported even if we don't see parasites, and we report white or red blood cells. The charcoal-laden crystals, as that picture I showed you way at the beginning, these are formed when the eosinophils break down in the intestine. You can see them in the stool, you can see them in the sputum of people that have parasitic diseases. But they can be misleading because eosinophils, if you've learned yet in hematology, also increase with asthma, allergic reactions, and with fungal infections. But it just is important to note those in case it is causing what we are looking for, those parasites. Okay, just quickly, the most common of this whole group that we're going to see in the United States is Giardia and Cryptosporidia. And we're not going to see those that often if our water supply is what it should be, is filtering the water. Because of that, some hospitals have dropped full O&P studies. They do not allow the doctor to order a full O&P unless the patient is severely immunocompromised, has traveled to a foreign country, or is just not getting better. Then they can order the full O&P. In the meantime, what they do is order a Giardia Cryptosporidium antigen test. It's a very quick, you take the stool, you treat it with a liquid, you drop it in the little cartridge, you drop a reagent, it migrates through like some other tests we've done, and it gives you a color change reaction. Doctors, though, are very demanding. A lot of doctors will not let hospitals give up doing that whole O&P, except most of them come back negative. I went to a conference that talked about that, and we actually did a study. We normally didn't see the results. O&Ps got sent out, and we never knew or cared what happened. I worked with our send-out department at RMC to see what actually came back. In a 6-month time, we had a Giardia or two, and maybe one crypto, and maybe one other parasite, and that was it. So I tried to talk to my pathologist, but the doctors are, of course, are more convincing than I was on whether to do that. But a lot of hospitals do that rapid Giardia crypto test that you will do in the lab. So that's our takeaway from all of this on what you're actually going to do.